Thank you very much also for the introduction. And I would also like to start by thanking the organizers of this conference for inviting me to give this talk. So my name is Alessandra Luchini, and I am the instrument scientist for one of the ESS reflectometer, STIA, which is a project mainly led by the Paul Scherrer Institute in Switzerland. But I'm not here today to talk about STIA, but rather about my research activities, which focus on the production and characterization of supported lipid bilayers with oriented membrane proteins. So as we also heard this morning, biological membranes are these very complex systems, mainly composed of hundreds of different lipid species and various and different membrane proteins. And they are often too complex to be directly studied with biophysical methods. Therefore, it is quite common to use instead model systems, such as a supported lipid bilayer that you can see here on the bottom, that are composed of these two layers of lipids deposited on a solid substrate. And you can actually prepare the supported bilayer with a highly variable lipid composition and combine many different surface methods to characterize both their structure and dynamics. And the main goal in the end is to extract physicochemical properties that are most likely to be similar in the corresponding native membrane. A bit more appealing are supported bilayer, which are not composed just of lipids, but they also contain membrane proteins. And if you have a membrane protein that has an asymmetric structure, such as the one represented here in the slide with one large extramembrane domain, it is quite desirable to have all of them oriented the same way in the bilayer, so all of them exposing the extramembrane domain towards the bulk solvent. So in this presentation, I will show you a method that we have recently introduced to prepare exactly this kind of systems. And this method is actually based on the use of peptide discs. Peptide discs are small discoidal particles which contain an inner core composed of lipids where you can actually incorporate your membrane protein of interest. And they are stabilized in solution by this red belt here that is composed of self-assembled peptide molecules. You can actually inject the peptide disc on a hydrophilic support and they will spontaneously absorb on the support surface. And because this peptide behaves a little bit like a detergent, so the peptide assembled to the lipid is actually in equilibrium with free peptide in solution, you can just remove the peptide by rinsing the surface with fresh buffer solution. And the initial patches of bilayer that are formed will then fuse and eventually form a continuous supported bilayer. Now, if you have a membrane protein incorporated in your peptide disc, because they are so small, they can approach the substrate surface with different orientation. And in the end, the orientation where the lipid head groups are closer to the substrate surface is the most favorable one. And this is why they will spontaneously orient all your membrane proteins in the same way. You can then characterize this system with different surface sensitive methods. Here you can see the one that we have directly used, but there could be plenty more. And today I'm going to show you some of the results we collected first on bilayers prepared just with phospholipids or phospholipid mixtures, and then some examples of membrane proteins. So we started with the classic reference sample that is POPC. It's a very common phospholipid, and you can see the structure down here. So it has a polar head group and two hydrophobic acyl chain. We initially collected QCMD data, and here you can see the results of the measurements. So we start at 0 with the calibration value of the sensor, and then as soon as we inject the peptide disc, the frequency shift decreases rapidly, and after buffer rinsing, we have a stable value of -25 Hertz. That is the characteristic value for a supported lipid bilayer. However, it was only after we collected neutral reflectivity data that we could indeed confirm the successful formation of a bilayer. And here you can see the scattering length density profile. To analyze this data, we used a pretty standard approach. So we represented our system as composed of two outer layers of head groups and one inner layer of acyl chain. And the structural parameters that we obtained were in good agreement with what was expected for a POPC bilayer. We also collected time-resolved infrared data to investigate the mechanism through which the peptide disk formed is supported by layer. And in the spectra, you can see here on the left side, the characteristic signal of the lipids. And on the right side, you can see the amide 1 band that is characteristic of the peptide. So you can see that the intensity of the bands increase quite rapidly during the injection of the peptide disk. And then the amide 1 bands start decreasing over time when we rinse it with a buffer solution until it reached the baseline value. And therefore, we came up with this schematic representation of the process where the peptide disk initially fuse, forming larger bilayer patches, and then the buffer rinsing step indeed promotes the removal of the peptide. We also tested different phospholipid mixtures that are quite commonly used to mimic mammalian membranes. So we use mixtures of POPC and POPS. POPS is an anionic phospholipid, so with a negatively charged head group, but also mixtures of POPC and POPE and POPC and cholesterol. And in all cases, we confirmed the formation of a supported lipid bilayer with the expected structure. We then decided to go to an even more complicated level, and in collaboration with ILL, we tested hydrogenous and deuterated phospholipid extract, which were produced from the yeast Pichia pastoris. And on the right side here, you can see that now these mixtures indeed are composed of a quite large variety of both head groups and different acyl chains. Despite the complexity of this phospholipid mixture, we could still prepare our peptide disk, and the data that you can see here show the successful formation of bilayers, both with the hydrogenous and the deuterated mixture. And there is actually a poster, I think presented by Giovanna, where you can learn more about these very interesting phospholipid mixtures and their application. But enough about lipids. I would like to talk more about membrane proteins. We have so far studied 3 different membrane proteins. The first one that we studied is called Core A. It is a bacterial membrane protein and a homopentamer. So it is actually composed of 5 identical units, and it has therefore a quite rigid structure. It is also a well-characterized protein, so the structure is known and it can produce relatively easily. So we decided to use this protein as a model system, as a test for our method. We then moved to a more biologically interesting membrane protein called tissue factor. This is a mammalian membrane protein that has a quite fundamental biological function as it initiates the blood coagulation process. And while the extramembrane domain of this protein was pretty well characterized because it can be expressed and produced as a soluble protein, there were so far no information about the full length structure of the protein in a lipid membrane. And this was actually what we were interested in investigating further. And then finally, more recently, we started investigating the structure of this other membrane protein that is called VAMP2. It is once again a mammalian membrane protein that is highly abundant in the brain and has a fundamental role in neuronal communication. Now, if you look at the molecular weight of this 3-membrane protein, you can see that we started with something quite large and rigid, and then we moved to something that is really small. And this was because we wanted, first of all, to test if our method would also work with the small membrane proteins. And as I explained for tissue factor and VAMP2, there were also biological reasons for investigating these 2 proteins. So I will first start by showing the data we collected on Core A. So once again, we started by testing our method with the QCMD. So again, we start here with the calibration value of the sensor. Then we injected our Core A loaded peptide disk and observed a very rapid decrease of the frequency shift. And after buffer rinsing, we got to a value that is about -70 Hertz. That is considerably smaller than the -25 that was recorded for a pure lipid bilayer. So already from this measurement, we had the indication that the heavier layer was deposited on the sensor surface. However, once again, only after we collected neutron reflectivity data, we could confirm the structure of our sample. And down here, you can see the data that we collected. I hope you can appreciate that because of the large size of this protein, we now have quite some fringes in the data that are not that common when you work with a single supported lipid bilayer. When we analyzed the data in the model, we added an external layer representing the extramembrane domain of the protein, and we also took into account of the transmembrane domain of the protein along the acyl chain. So from the analysis of the data, we first of all could confirm that core A is indeed present in the bilayer, that all the core A molecules are oriented the same way. So they all expose the large extramembrane domain towards the bulk solvent. And we also measured the thickness of this extramembrane domain, which resulted to be in good agreement with the crystal structure of this protein that, as I mentioned, was known. Now moving to the second case, the case of tissue factor. The protocol was the same. So we started by preparing our TF-loaded peptide discs and by initially performing QCMD measurements. So once again, we started with a value of the calibration of the sensor corresponding to 0 up here, and then injected our peptide discs, and this time observed a final frequency shift value, which was about -35 Hertz, which is larger than what was observed before for core A, but still considerably smaller than the value recorded for the pure lipid bilayer. And this is in a way was expected since TF has a much smaller molecular weight than core A. Then again, we collected our neutral reflectivity data that you can see down here. And perhaps you can appreciate that there are now two different sets of fitting curves on top of our experimental data. This is because we used two different models or compared to different models with respect to our data. One model was exactly the same as I just explained in the case of core A. So if you want, it's a simple layered model. And the other model that was used was actually based on MD simulations. So in parallel to these experiments, we performed indeed MD simulations, and from the structural model that was provided from MD, we calculated first the corresponding scattering length density profile, and then the reflectivity curve. So the main results that we obtained here were that, first of all, once again, we could confirm the presence of the TF molecules in the bilayer and their orientation with the extramembrane domain exposed towards the bulk solvent. By having the support of DMD data, we were also able to see that the extramembrane domain is actually tilted towards the lipid head group surface, and this confirmation is actually favored by the presence of PS lipid in the bilayer. We also then, I'm not going to show you data on this because of lack of time, but we also then observed that this confirmation is actually quite important for this protein to bind the soluble coagulation factor 7A and is therefore fundamental for the biological function of this membrane protein. And now getting to the last case, the case of VAMP2. This was a much more challenging protein to work with due to its small size. So once again, we started by collecting our QCMD data. And the overall data looked quite similar to tissue factor. Perhaps the frequency shift here was slightly larger than in the previous case, but this was also reasonable considering that VAMP2 has a smaller molecular weight than tissue factor. However, when we collected our neutron reflectivity data, as you can probably see in these plots, the data trend was quite similar to a pure lipid bilayer. So we started wondering whether there was really any protein in our bilayer or not. And we then analyzed our data and found out that there is some protein, but it's a very small amount. So if, for example, we take the volume fraction of the extramembrane domain of this protein, that would be something smaller than 10%, which then makes quite difficult to really extract any reliable structural information on this protein. We can still see that there is some protein in the bilayer. This is also confirmed by the fact that we have a higher scattering length density in the acyl chain region compared to pure lipid. But just based on this data, we can't say really much about the protein structure. We are now thinking of perhaps trying to implement in our model independent information that are available on this protein, since a few years ago there was an NMR structure published. Even if neutron reflectometry cannot really help us saying much more about the protein structure, we can still say something about its orientation. And this is because if we consider the opposite orientation compared to what is shown here, so if the, for example, the extramembrane domain was oriented in the opposite way, so towards the support surface, and even if we squeeze it really tight to the bilayer surface and partially penetrating the head group, we should still have a low density protein layer here in between the substrate and the bilayer of at least some 10 angstroms of thickness, which would in the end produce quite a different data trend compared to what we obtained. So at least we can say that neutron reflectivity supports the formation of a bilayer with the expected orientation of the protein. But to really confirm that the VAMP2 protein was indeed present in our bilayer, we collected again our FTIR data. And these are again time resolved. So we monitored the formation of the bilayer with the VAMP2 incorporated in the peptide disk. And once again, you can see here on the left side, the characteristic absorption band for the lipids, which increase very rapidly in time and then remain stable during buffer rinsing. And on the right side, you can see our amide 1 band that this time is much more intense compared to what was obtained in the case of a pure phospholipid bilayer. And this is because this time we not only have the peptide contributing to it, but we also have the VAMP2 protein. So the intensity of this band increased quite rapidly in time. Then we started rinsing to remove our peptide. So it started decreasing, but instead, of course, of going back to the baseline value, it remained it remained stable to a certain intensity. And this is where we can see that it's mainly VAMP2 contributing to this band. So then we zoomed in and looked more closely at this band that you can see in this zoomed plot here, and notice that its shape is actually quite asymmetric, and it can be then analyzed considering two contributions. One contribution corresponding to an alpha helical structure, and the second contribution corresponding to a beta sheet structure. And this is actually in agreement with a paper that was already published on VAMP2 and FTIR data, although in this previous study, only the transmembrane domain of VAMP2 was used, and it was in a lipid multilayer system and not in a single bilayer. So I hope that with this presentation, I have convinced you that the peptide discs are indeed a very interesting system when it comes to form supported lipid bilayer with a highly variable lipid composition, including different synthetic phospholipid mixtures, but also more complex natural lipid extracts. You can incorporate in this bilayer membrane proteins with a controlled orientation, and it is compatible with different surface sensitive methods and membrane protein of different sizes. Although if you are mainly interested in neutron reflectometry experiments, perhaps when you are approaching the VAMP2 sizes, that might be a little bit of the limit. This supported bilayer can be used to characterize the structure of your lipid membrane or of your membrane protein, but they can also be used as platform to, for example, investigate binding processes that involve the extramembrane domain of this membrane protein. Looking forward, we are actually quite interested in keep developing this method and in trying to develop new peptide variants that could be used to, for example, improve the formation of the bilayer and removal from the lipids, but also make more easier to detect the protein concentration. And about this, I just have one more slide about the new peptide that we started working with that we called M18A. This is because the standard peptide used to form this peptide disk is called 18A, and you can see its sequence down here. And what we did was actually replace this tryptophan with the glutamine, and the consequence of that is that the peptide now have no absorption at 280 nanometer. So during our size exclusion purification of the peptide disk, we can see that the pure peptide disk, so those made just of lipids, they only absorb at 214. Whereas when we have also a membrane protein incorporated, we can follow the peptide, monitoring the 214 absorption, and follow just the membrane protein for with the 280 absorption. And this was a case of the VAM2 loaded peptide disk, but this will be the same for other membrane proteins, which then means that one can also use the absorption of these wavelengths to directly quantify the concentration of membrane proteins in the peptide disk. With this, I would like to thank all the people that contributed to this work, and in particular, Louise Arlet and the former members of our group at University of Copenhagen. I was also part of that group for quite some years, and this is where this entire project started. I would also like to thank Federica Sebastiani and Marité Cardenas from Malmo University. That's where I collected the QCMD data, and in particular, Federica provided a lot of support for the collection of the infrared spectroscopy data. The neutron data that I've shown you were collected at ICIS and ILL, and I would like particularly to thank Giovanna and her group for the natural lipids. And then finally on this slide here, I hope you can see the picture of our Selene Guide 1, the first component of the STI instrument that was installed this summer at ESS. The picture is already quite outdated because the surrounding building has evolved quite fast, and it's looking much more different already now. And we are looking forward to continue with the installation of our instrument during the spring and the summer. And I would like to thank you all for your attention.